pretend Boyd's here and Jack's here. So today our video is going to be talking about DNA and RNA and what those things do in this big whole concept that we're learning about. I hope this video is helpful and brings light onto a tricky concept that I hope all of you understand by the end of this video.
What is DNA made of? So DNA is made up of nucleotides, and each nucleotide consists of a nitrogenous base, a deoxyribose sugar, and a phosphate. So the four nitrogenous bases are adenine, thymine, guanine, and cytosine. These are the rungs of the ladder, and they are hydrogen bonds that connect each nucleotide together. So, for example, this is adenine, and it will be paired with thymine. This will be guanine, and this will be paired with cytosine. One side of DNA is a pattern of nucleotides that match up with the other side. So DNA looks like a double helix or a twisted ladder when it's in its original form. On DNA, the base pairs that match up are adenine and thymine, and guanine and cytosine. So on the DNA, it would be A and T and G and C. So purines are two carbon nitrogen ring bases, and adenine and guanine are purines, and pyrimidines are one carbon nitrogen ring bases, and thymine and cytosine are pyrimidines.
Hi, I'm Jack Eames, and I'm here to talk about the replication process that occurs in DNA. So here, as you saw me draw, I have four basic steps that concludes DNA replication. So the first step is an enzyme called DNA gyrase kind of makes a cut right in here, which enables it to start to flatten out. The enzyme that is actually responsible for unwinding the double helix formation, as Boyd just talked about, is called helicase, and it kind of just quote-unquote treads over the double helix to flatten it out. Okay, so once that's done, the hydrogen bonds broken, which are over here. I didn't diagram them due to the fact that they're being broken. So the hydrogen bonds are going to be broken, which is going to enable the strip to be kind of peeled apart. And next comes in the SSB, or the single-strand binding proteins, which keep these two strands temporarily pushed away because adenine and thymine and guanine and yours still naturally.
He meant thymine.
Okay, so after SSB is holding off these two nucleotides from bonding, this enzyme called DNA polymerase walks along the flattened out double helix, pairing the new nucleotides, so adenine with thymine, cytosine with guanine, and reversible as well. So once all of them are matched up and the DNA is almost finished, an enzyme called DNA ligase kind of finishes up and seals all the stuff together. And then once that's done, the new strand will wind itself back up, and then you have a new strand of DNA. Okay, so we have our new DNA strand. After I've covered the actual process of replication, I'm going to go into a bit more detail. So there's one thing called the parent strand, which is the original DNA molecule that we have here. The parent strand is half of the new strand that we have here, and half of it would be the daughter. So this half, so you could say that this half would be the parent strand, this half gets removed, and then we're going to be attached by the new daughter strand. So the new daughter strand is identical to the DNA, but it's not the same. It's not the mother. And while all this is happening, all of this DNA replication and all the DNA is contained in chromosomes, which can be found in the nucleus. Okay, so overall, DNA replication is a pretty cool process. The real importance of it is if we didn't have DNA cells that wouldn't replicate, we wouldn't be here today. So that's the only reason why things are made. DNA is what makes up our body. We're made up of DNA. So without replicating, we either start out as just one little guy here. Everything needs to DNA, replicate. Everything from plants to humans to even dinosaurs. Everything replicated. Without replication, we would all die. So yeah, that has been my covering of DNA replication.
So transcription is the process when a DNA strand is split apart, then an mRNA strand comes in and copies one of the DNA strands, and once that happens, the mRNA strand detaches and leaves the nucleus where transcription happens, and then as soon as that happens, the DNA strands come back together into a double helix.
Many people ask the difference between DNA and RNA. DNA stands for deoxyribonucleic acid. RNA stands for ribonucleic acid. In DNA, there is one less oxygen than in RNA. The three types of RNA is mRNA, tRNA, and rRNA. There's only one type of DNA, and that is DNA.
So, the reason why transcription is such an important role is because without transcription, the mRNA could not decode the amino acids, resulting in the tRNA could not transfer the amino acids to the ribosome, and the ribosome will not be able to conduct protein synthesis, resulting in no proteins in our body, and we would all die.
Transcription happens in the nucleus.
Translation. So translation occurs in the ribosome. And so translation is when an mRNA strand with a codon on it comes into the ribosome from the nucleus, and a tRNA strand comes in with amino acids attached to it. And so the job of the ribosome is to decode the amino acids. Decodes the mRNA to make a sequence of amino acid chains.
The genetic code, which DNA stores the genetic information, consists of codons of 3 nucleotides. The functional segments of DNA, which code the transfer of genetic information, are called genes. The ribonucleic acid, or RNA, that is directly involved in the transcription of the pattern of bases from the DNA to provide a blueprint for the construction of proteins, is called messenger RNA, or mRNA. The pattern for protein synthesis is then read and translated into the language of amino acids for protein construction with the help of tRNA, which is known as transfer RNA.
Proteins are a huge part in translation. Proteins are polymers, or specifically known as polypeptides, and in a sequence, they form various amino acids. Protein plays a big part in translation because translation is the final step from DNA to protein, and the synthesis of proteins is directed by the mRNA template, which is messenger RNA, and during the translation. The whole thing of translation, amino acids link together to form a polypeptide chain. They will later be folded during that process into protein. So that protein is then made, and then there is the ribosome, which is the cellular structure responsible for protein synthesis, and that plays a big part in translation too. It consists of 2 different subunits, one small and one large, and it's built of rRNA and proteins. So proteins, as you can tell, are very big in this process.
These 3 stand for ribosomal RNA, messenger RNA, and transfer RNA.